Histology is a branch of biology that studies the structure and composition of human body tissues, or other organisms, using a microscope. The term histology comes from the Greek, where histo means tissue and logos means study or science. Histology is very important in medicine because it allows a deep understanding of how cells and tissues of the body function and how changes at the microscopic level can contribute to disease. In detail, histology studies the composition of cells and tissues in the body, both in terms of structure, function, and changes that occur in pathological conditions. Types of tissue in the human body. In general, human body tissue is divided into 4 main types, namely, A. Epithelial tissue. Epithelial tissue consists of layers of cells that form the surface of the body and body cavities, and limit organs and body channels. The epithelium functions in protection, secretion, absorption, and excretion. The function of epithelial tissue, namely, protecting the body from pathogens, helping the absorption of nutrients, for example in the intestines, secretion, for example glands, and excretion of waste products, for example in the kidneys. Examples: squamous epithelium, flat, in the skin, columnar epithelium in the digestive tract, and cuboidal epithelium in the thyroid gland. The structure of epithelial tissue, namely, usually consists of 1 or more layers of dense cells and does not contain blood vessels. Nutrients are obtained through diffusion from the underlying tissue. B. Connective tissue, conjunctive, namely, connective tissue consisting of cells dispersed in an extracellular matrix consisting of fibers and basic substances. This tissue functions to connect, support, and provide structure to other parts of the body. The function of connective tissue, namely, providing structural support for body organs, storing energy in the form of fat, and playing a role in the immune system. Examples: loose connective tissue, such as adipose tissue, dense connective tissue, such as tendons and ligaments, bones, and blood. The structure of connective tissue, namely, consists of 2 main components, cells and an extracellular matrix. This matrix consists of collagen and elastin fibers, as well as basic substances that provide strength and flexibility. C. Muscle tissue. The description is, muscle tissue consists of cells that can contract and produce body movement. There are 3 types of muscle tissue: skeletal muscle, cardiac muscle, and smooth muscle. The function of muscle tissue allows body movement, movement of internal organs, e.g., peristalsis in the intestines, and maintains body posture. Examples: skeletal muscle, connected to bones and allows voluntary movement, cardiac muscle, found in the heart and regulates heartbeat. Smooth muscle, found in internal organs such as the intestines and blood vessels. Structure of muscle tissue, namely, muscle cells have long fibers that contain the contractile proteins actin and myosin, which allow muscle contraction. In skeletal muscle, muscle fibers have a pattern of light and dark lines, transverse lines, while smooth muscle does not have these lines. D. Nervous tissue. Description of the nervous tissue, namely, nervous tissue consists of neurons and glial cells. Neurons are cells that transmit electrical signals, while glial cells support and protect neurons. Functions of the nervous tissue, namely, controlling and coordinating body activities, including processing sensory information and sending signals to muscles and organs. Examples: motor neurons, sensory neurons, and glial cells such as astrocytes and oligodendrocytes. Structure of the nervous tissue, namely, neurons have three main parts: cell body, which contains the nucleus, dendrites, which receive signals, and axons, which transmit signals. 2. Techniques in histology studies. Histology requires the use of certain techniques to prepare tissue samples for viewing under a microscope. Some of the main techniques in histology include sample preparation, tissue staining, microscopy. A. Sample preparation, consisting of 1. Fixation, namely, the tissue sample is first fixed with chemicals, such as formalin, to prevent cell damage or decay. 2. Dehydration, namely, the tissue sample then undergoes a dehydration process, where the water in the tissue is replaced with alcohol. 3. Insertion, namely, the dehydrated tissue sample is inserted into a hard medium, such as paraffin or resin, to facilitate thin cutting. 4. Cutting, namely, the hard tissue sample is then cut with a microtome into thin slices that can be seen under a microscope. B. Tissue staining, which includes hematoxylin and eosin, H&E. Specific staining 1. Hematoxylin and eosin, H&E, namely, the most common staining used to highlight the difference between the nucleus, which will be blue or purple with hematoxylin, and the cytoplasm, which will be pink with eosin. 2. Specific staining, namely, several stains can be used to highlight specific tissue components, such as silver staining for collagen fibers or PA staining for carbohydrates. C. Microscopy, which includes light microscope and electron microscope. 1. Light microscope, namely, this microscope is used to observe tissue that has been prepared and stained. Light microscopes allow visualization of microscopic structures at the cellular level. 2. Electron microscope, namely, this microscope provides higher resolution and is used to view more detailed structures, such as cell organelles and subcellular components. 3. Applications of histology in medicine and research. Histology is very important in various fields of medicine and research, including a. Disease diagnosis. Histology is used to diagnose various diseases, including cancer, infections, and autoimmune disorders. Tissue samples taken through biopsies can be analyzed to detect changes at the cellular or tissue level, such as abnormal cell growth or changes in cell structure. Example: histopathology examination to diagnose breast cancer, or to determine the presence of infection in body tissues. B. Physiology and cell biology research. Histology is also used to study the physiology of the human body and the processes of cellular biology. This research is important to understand how cells and tissues work under normal conditions and in pathological conditions. Example: research on the role of immune cells in inflammation or how muscle tissue regenerates after injury. C. Drug development. Histology is used in drug development to study how drugs can affect certain tissues or organs. This is important in ensuring the safety and effectiveness of drugs. Example: histological tests to evaluate the impact of drug side effects on body tissues, such as liver or kidney damage. IV. Diseases associated with tissue changes. Histology also helps in understanding changes in body tissues that occur during disease. Here are some examples of diseases that can be recognized through histological changes, namely, a. Cancer. Namely, cancer can be recognized through histological examination, which shows abnormal cells with uncontrolled division characteristics. Types of cancer, such as carcinoma or sarcoma, can often be diagnosed by observing the differences between normal and cancerous tissue cells. B. Infection, namely, microorganism infection can damage the structure of body tissue. Histological examination can show signs of infection, such as inflammation or the presence of microorganisms in body tissue. C. Autoimmune diseases, namely, in autoimmune diseases, the immune system attacks the body's own tissues. Histological changes in body tissues can provide clues about the type and severity of autoimmune disease. Conclusion: histology is a very important science in understanding the structure and function of human body tissues. Using microscopy and staining techniques, histology allows us to see and analyze how cells and tissues work, and detect pathological changes that occur at the cellular level. The applications of histology in medicine are very broad, ranging from disease diagnosis to drug development research, making it a key component in understanding human health and disease.